In this video, we're going to go through the structures of a nucleus and the roles of the different parts that make up the nucleus. So what we're looking at on the screen in front of us at the moment is a nucleus made up of a double membrane, okay? So you can see each of these little sections here is basically representing a cross-section through that double membrane. Now remember, the nucleus is completely spherical, so it's three-dimensional like the shape of the sun, but dotted with lots of little holes which are called pores, okay? So these little parts that we can see here are actually the nuclear pores. Now there's over 3,000 of these nuclear pores that make up the nucleus. Now the first and probably the most important function of the nucleus is the fact that it's there to contain the DNA. It's like a prison for DNA, okay? You don't want your DNA to be lost. It's really, really important, okay? It's made up of these chemical bases that basically is the code for the amino acids that are then going to make proteins in the cell, okay? So you need to keep your DNA inside the nucleus, okay? So in here, it contains DNA. DNA is usually in the shape of chromosomes, which we'll talk about in a moment, okay? What else is found inside of our nucleus? Okay, we've also got these things called a nucleolus, okay? So they're like small, dark, spherical structures that you'll see inside of the nucleus. There could be one, there could be multiple nucleolus, okay, like you can see here. So we've got 3 on this image here. So we've got our nuclear envelope made up of a double membrane, okay? It contains nuclear pores, which are very small. The whole point of the nuclear pores is to allow large molecules to move out. Now, not large molecules as big as the DNA, but large molecules such as, for example, messenger RNA. Now, messenger RNA is something you're going to learn more about later on, okay? Basically, when your DNA needs to make a protein, okay, from a specific section of DNA, which is called a gene. So when the gene needs to code for a protein, what will happen is the messenger RNA will basically pair up with any 3 bases that are complementary to it, okay? The messenger RNA, unlike the DNA, is single-stranded, and the messenger RNA therefore is small enough to move out of the nucleus. Okay, so it will create like a complementary copy of that DNA in a strand of messenger RNA. That's a single strand, and then the messenger RNA that we can see is small enough to move out of the nucleus and can go into the cytoplasm, where it will attach to a ribosome in the cytoplasm. Okay, so here's our messenger RNA here attached to that ribosome, and the ribosome is going to read that code, and along with the help of tRNA, which we have here, tRNA, sorry about the writing, it's not very easy on here, it will code for amino acids, which will then make up a protein. Now, we'll learn more about that in transcription and translation later on. But basically, the pores are small enough to allow large molecules out, like messenger RNA, but small enough also to make sure that the DNA, which is an even bigger molecule, stays inside the nucleus. Okay, now the other role of the nucleus is really to help compartmentalize any chemical reactions that are going on inside the nucleus. Now, what does that mean? Now, that's the same as really any organelle. You want to compartmentalize the chemical reactions. So you want to keep the reactions and the pH that's inside the nucleus from interfering with anything else that's happening inside the cell. Okay, so it's like closing off everything in a cupboard, making sure it doesn't affect anything else happening inside the cell. So the other thing we have inside the nucleus, okay, is basically this whole area that we can see inside the nucleus contains something called nucleoplasm. And nucleoplasm is a jelly-like substance, a bit like cytoplasm, but it's only found inside the nucleus, okay? And that's really the bulk of the nucleus. Now, This nucleus and the nuclear membrane is really, really important in the cell. It's going to control what happens inside the cell, and it will do that through the production of proteins. Okay, let's have a look. Slide. Now, if we look at the DNA that's inside the nucleus, this is an example of DNA. DNA is found, as we can see here in the text, in a double helix. Okay, so it's two strands like this, so two individual strands that are then wrapped around each other, like you can see here, okay, and they form this double helix structure. Now, what else is involved with the DNA in order to make the chromosomes, okay? Now, if we think of our DNA like this, DNA inside a eukaryotic cell is wrapped around proteins. I'm just going to draw some of these proteins in here. So these proteins are called histone proteins. Histone. So histone proteins basically allow the DNA to coil up around it, forming a dense supercoiled structure of DNA. Okay, now this is what allows over 2 meters of DNA, for example, to actually fit inside one nucleus. Now that is not an unsubstantial amount of DNA. Okay, now by coiling up around these histone proteins, the DNA is able to fit inside the nucleus, but also you'll see in later information it's going to learn that they have a role in helping to switch the DNA on and off for particular genes. Okay, so that means turning genes on to allow a protein to be made, or turning genes off to stop a protein being made. Okay, now only eukaryotic cells have the histone proteins. If it's a prokaryotic cell, it does not contain a nucleus, first of all, and also its DNA is what we call naked DNA. Now that means in a prokaryote there are no histone proteins. Okay, so naked DNA would not have any histone proteins attached to it, but DNA in eukaryotic cells does have histone proteins attached to it. Okay, we also call this linear DNA because although it's a double helix like we've seen, okay, the two ends are separate. So we've got an end here and we've got an end here. They don't go back to join back on themselves like we find in a prokaryote with a circular DNA. We've got two clear, distinct ends. Now, once the DNA is supercoiled, it will actually appear in the nucleus as a chromosome, and this only happens just before cell division. So if we have a look at a chromosome, you'd have learnt the structure at GCSE. It's actually made up of two chromatids so two chromatids joined together in the middle by a centromere. Okay, and this, if you imagine, is made up of loads of coiled up DNA. So it's in its double helix and then it's coiled around histoproteins into this structure to make sure it's really, really condensed nice and tightly. Now humans have 46 chromosomes, that's 23 pairs of chromosomes. The number of chromosomes you have does not make you a certain level of intelligence. In fact, actually loads of other animals also have 46 chromosomes as well. Including marmosets, I believe, and the meadow vole. And something that has the largest number of chromosomes, which is the adder's tongue, is just a plant. So it doesn't matter how many chromosomes that the organism has, that doesn't make it a certain level of intelligence. Okay. Now, the nucleus contains the nucleolus, and we can see 3 nucleoluses here present. Okay. It appears as a dark spherical body inside the nucleus. Now, the role of the nucleolus is to manufacture ribosomal RNA, so little r, capital R, N, A. And this is needed in order to make ribosomes. So, not only do they manufacture RNA, they also assemble, as you can see here, they assemble the ribosomes from 2 subunits. That's a smaller subunit and a larger subunit that are joined together in order to make an individual ribosome. Now, these ribosomes are actually ATS ribosomes in a eukaryotic cell. Okay, the ribosomes will leave the nucleus and they'll either become a membrane-bound ribosome, in which case they'll be attached to the rough endoplasmic reticulum, which is actually continuous with the outer membrane of the nucleus, like this. Okay, and those ribosomes that are bound will appear as dots on the rough endoplasmic reticulum, like so. Or they will be what we call free ribosomes in the cytoplasm. So they won't be bound to a membrane, they're just free inside the cytoplasm. Okay, and both ribosomes do the same job, but they produce proteins that are going to go to different places. Now, I always teach fibo. Now, it might sound silly, but let me just find a way of writing it up here that I don't get in the way of the camera. Fibo. So what this means is free ribosomes make proteins that stay in the cell, which is what the I is for, and bound ribosomes make proteins that go out of the cell.